All right, welcome to The Explainer. Today, we are cracking open one of the most important chapters from your NCERT syllabus, Biotechnology Principles and Processes. We're going to break it all down into exactly what you need to nail those questions for your NEET prep.
So let's just jump right in with a pretty wild question. How in the world can you tell a simple little bacterium to start making something as complicated as human insulin? I know it sounds like something out of a sci-fi movie, right? But it's not. The answer lies in the incredible power of genetic engineering.
And to get our heads around that, here's our game plan. We'll start with the basics. What even is biotechnology? Then we'll look at the two principles it's built on, pop open the genetic engineer's toolkit, see how the whole rDNA assembly line works, and finally, follow the journey from the lab all the way to the market.
Okay, first up, let's get our definitions straight. What exactly is biotechnology?
You might think this is some super modern thing, but people have been using biotechnology for thousands of years. Anytime you make bread, curds, or wine, that's biotech. But the modern definition, the one from the European Federation of Biotechnology, is way broader. It's really about mixing natural science with the living things, organisms, cells, even molecules, to create products and services we can use.
All right, section 2. Now, this is important. All of modern biotechnology, and I mean all of it, really stands on two fundamental pillars. These are the core techniques you have to understand.
On one hand, you've got genetic engineering. This is where the magic happens. It's all about directly changing an organism's DNA or RNA to give it brand new characteristics. On the other hand, you've got bioprocess engineering. This is the muscle. It's about creating these perfectly sterile, controlled environments so we can grow trillions of our modified cells to manufacture things like vaccines or antibiotics on a massive scale. One's the blueprint, the other is the factory.
So for section 3, let's talk about the fun part, the genetic engineering toolkit. Because to actually do all this cool genetic engineering, you need the right set of tools. So let's open up that toolbox and see what's inside.
Here are the absolute essentials, the non-negotiables, for making what we call recombinant DNA. We've got restriction enzymes, which act like tiny, precise scissors. We have cloning vectors, which are our delivery trucks. We've got DNA ligase, which is our molecular superglue. And finally, the competent host, which is the factory where all the production happens. Let's start with those molecular scissors, the restriction enzymes. And the precision here is just incredible. These enzymes float along a DNA strand and only make a cut when they find a very specific sequence of letters, what we call a palindromic recognition sequence. For example, the enzyme EcoRI is always on the lookout for GAATC. It sees anything else, it just keeps on moving. And this image shows you exactly how that works. See how EcoRI doesn't just slice straight through? It makes a staggered cut between the G and the A on both strands. This leaves these little single-stranded overhangs, and we call these sticky ends for a great reason. They're just waiting to pair up and form hydrogen bonds with another piece of DNA that's been cut with the very same enzyme. This is the whole key to pasting different pieces of DNA together.
Okay, so you've used your scissors to cut out your gene. Now what? You need a way to get it into your factory, the host cell, and that's where our delivery truck comes in, the cloning vector. Most of the time, these are plasmids, these little circular pieces of DNA that bacteria naturally have, and they can copy themselves independently.
And here's a famous one, a true classic, PBR322. This plasmid is like the workhorse of molecular biology labs all over the world. It's been perfectly engineered for one job, cloning. So what exactly makes a vector like this so perfect for the task?
Okay, everyone, lean in close for this one because this is a huge topic for your exams. A good cloning vector must have 3 key features. Number 1, an ORI, or origin of replication. That's the start signal that tells the host cell, hey, copy me. Number 2, a selectable marker. This is usually an antibiotic resistance gene, and it's a clever trick that helps us find which cells actually picked up our plasmid. And number 3, cloning sites. These are specific, unique spots where our restriction enzyme can make a clean cut to insert our gene. So for section 4, we've got our principles, we've got our tools. Now let's see how it all comes together. Let's walk through the rDNA assembly line, step by step.
When you look at it, the process is actually very logical. First, you've got to get pure DNA to work with. Then, you use your restriction enzyme scissors to chop it up and get the fragment you want. After that, you use your ligase glue to paste that gene into your vector. Next up is transformation, which is just the process of getting the host cell to take in your new recombinant vector. And finally, you let that host cell grow and divide, amplifying your gene of interest into millions of copies.
Now, about that amplification step. To make millions or even billions of copies of just one specific piece of DNA, scientists have an amazingly powerful technique. You've almost definitely heard of it. It's called the polymerase chain reaction, or PCR.
PCR is really just a simple cycle of changing temperatures over and over again. It's a three-step dance. First, denaturation. You crank up the heat to separate the two DNA strands. Second, annealing. You cool things down so that little starter pieces called primers can bind to the DNA. And third, extension. A special heat-loving enzyme called Taq polymerase gets to work and builds the new strands. You repeat that cycle 30 or 40 times, and you can get over 1 billion copies from just one original molecule. It's incredible.
Okay, but wait a minute. We've used our enzymes to cut up a whole genome. How on earth do we sort through that mess and find the one specific little fragment we actually want? Well, for that, we have another brilliant technique called gel electrophoresis. I want you to think of this gel as a kind of molecular obstacle course. We load our DNA mix at one end and apply an electric current. Now, DNA has a negative charge, so it naturally starts moving toward the positive electrode. But here's the catch. The gel acts like a sieve, and the smaller DNA fragments can wiggle through it much faster than the big, bulky ones, so they separate by size. Then we stain the gel with a dye that sticks to DNA, and when we shine a UV light on it, bam! You see these beautiful glowing orange bands. We can then literally cut out the band we need, and we're good to go.
And that brings us to our final section, from lab bench to market. We've done all this amazing molecular work. We have our recombinant organism. But how do we go from a tiny sample in a lab to making enough of a product, say insulin, for millions of people? The answer is these things: bioreactors. These are basically giant, high-tech, super-controlled vats. They can be huge, holding hundreds or even thousands of liters. Inside, every single condition is perfectly optimized: the temperature, the pH, the oxygen levels, the nutrients, all to get our modified cells to grow like crazy and pump out our desired product.
But we're still not done. Once the bioreactor has done its job and produced all this material, it has to go through a crucial final phase called downstream processing. This is all the cleanup work. It involves carefully separating and purifying the product, adding preservatives to make it stable, and running tons of strict quality control tests to make absolutely sure it's pure, safe, and effective.
So there you have it. We've gone from the core principles to the amazing tools to the entire process from a single gene to a finished product. It's quite a journey, so I'll leave you with a final thought to chew on. Of all the incredible tools in this toolbox, the restriction enzymes, the plasmids, the PCR machines, which single discovery do you think was the real game changer? The one that truly unlocked the door to the modern age of biotechnology? Give it some thought. Thanks for joining me on The Explainer.